protein denaturation. So blood is not only involved in transportation, but it's also involved in homeostasis. And maintaining the pH is super important because if the pH falls out of the acceptable parameters, which is something like 7.35 to 7.45, something in there, the proteins in your blood will fall apart. Remember, protein shape, conformation, is heavily dependent upon environmental characteristics such as temperature and pH. So that's a super important thing to remember. Body temperature. So we talked about pH, proteins falling apart. Remember, and all the things you said were secondary, right? So clotting, are there some important clotting proteins? Yep, we talked about one in lab. You said infection. Are there important proteins involved in fighting off infection? Yes. So all that you said was kind of secondary, and the main concept there being that the proteins are all going to fall apart. Things are going to fall apart in the blood that your body needs. So pH is super important. Body temperature is also important, and the blood regulates this. Blood, how do you feel when you get super hot? Flushed. Blood kind of comes to the skin. You know that feeling when your blood comes to your skin? All the blood's kind of shunted towards the skin. Why? It's dropping off heat, right? It's dropping off that heat to try to cool the body down. When you sweat out of your skin, okay, so that's actually releasing heat. And whenever that sweat evaporates, it cools you down. So temperature is really important to that regulation and blood being homeostatic. Also, maintaining adequate fluid volumes. Those fluid volumes, when they drop too low, like let's say you cut off a limb, it sends your body into shock, right? So we have mechanisms in place to recognize that, and so that you can be conserved as much as possible. So those blood vessels at a place of injury, if you had lost a limb, would actually kind of close off, right, trying to conserve blood. So that fluid volume is super important. And that's a protective mechanism, so blood has the function of protection, preventing blood loss, preventing infection. So blood, mainly people think of gases, oxygen and carbon dioxide very often when they think of blood, and they think transportation is really quick or the first thing people think of. But we got to think about the overall picture. What all does blood do? So it has many functions to consider in the human body. Very important. And it is connective tissue. So if you recall, I've told you that one of the major things you learned in AMP1 that carries over is the terminology, but also the tissues. So every time we cover a system, I'm going to ask you about those tissues and what they do. And in this particular situation with blood, we're dealing with connective tissue. Does anybody remember what connective tissue begins with as embryonically? Mesenchyme. Very good, Alexis. Mesenchyme. To be connective tissue, you start out as mesenchyme. All connective tissue begins as mesenchyme. All right. Seen this picture before? Same picture that's in your lab manual, right? It is. We talked about today in lab briefly, what can you get out of blood? What can somebody know about you from your blood? Your entire genetic makeup. Also, you can be diagnosed by reading the blood, right? Many different things in blood. You could look at protein concentrations. You could look at white blood cell concentrations. You could look at red blood cell concentrations. You could look at hormone concentrations, right? You could also look at that genetic makeup. Your blood is an extremely personal thing, and it tells a lot of information about you. I told you earlier, wouldn't the health insurance companies like to have a vial of your blood? I bet you if they could get it or find your genetic makeup, they would buy it. They would purchase it. They may find some loophole to deny you health insurance or make a really high premium. I don't know, but they would like to have that information on you. It's currently illegal for them to require that information, so they can't make a judgment based upon your genetics. Just because you're predisposed to have cancer, does that mean you're going to have cancer or die from cancer? No, it doesn't. But we can look at the genetics to see high cholesterol. People are genetically predisposed to have high cholesterol. Their genetics says their cells are not going to have the receptors to take up as much cholesterol into the cell. What does an excessive cholesterol floating around in the blood do? It backs up in the arteries, makes it blood flow not as much, not as easy, so it's not a good thing. Atherosclerosis, right? So some people have really low cholesterol. You ever had that person that could eat eggs and bacon every day and still have low cholesterol? That person's not me, but they're genetically predisposed to have lots of receptors to uptake that cholesterol out of the blood. You hear people that are like, oh, I smoked cigarettes for 100 years. That's kind of rare, but yeah, it happens. Well, guess what? They may have had great genetics, above average genetics, that allowed them to have high regeneration in the lungs or whatever the case, but that's usually an anomaly. Obviously, research has proven over and over again that cigarette smoking is pretty much a death sentence if we continuously do it. You can stop smoking or quit smoking and reverse damage, right? Our bodies are very regenerative. They have very regenerative capabilities, but if you continue to do that and put your body through that, you're going to suffer. And from many different avenues, whether it be cancer or decreased lung capacity, any of those things. So smoking is definitely bad. But your blood tells a lot about you. When you go to the doctor, we give our blood pretty freely, correct? And you should at this point in time. You know, being a blood donor is a good thing. It's good for you. Did you know it was good for your body to donate blood? It is. It's very good. It basically is going to stimulate you to make more new blood, new fresh blood. Very fresh new red blood cells, make new white blood cells. It's going to stimulate your immune system, right? It's very good for you to donate blood. Getting rid of that pint will put you in the mode of making new. You're going to be a little tired right after giving. You've lost a pint of blood, right? Your energy may dip down, but it's going to go back up once your body starts that regeneration process. So you get a lot of blood, and there's lots of things that can be diagnosed or told about you. So one of the first things they do when you go to the doctor is they're going to take some what? Blood. They'll very often take urine too, right? Urine and blood, so those two things can tell a lot about a person. If you're pregnant, you're going to walk into your OBGYN and they're going to say, give me some urine, give me some blood. And basically they're going to run certain common tests to make sure everything's kind of where it needs to be. And this is what they're going to do. They're going to take your blood, and the picture is showing you what they're going to do. They'll take it, basically they put a little rubber apparatus, I don't know, the tourniquet, they usually put that off, and it keeps a good supply of blood here, and also makes it easy for them to draw the blood. So they'll draw your blood, and they're going to centrifuge it. So when they centrifuge your blood, it's going to spin it really fast, and it's going to separate by density. But it will be separated into these components, and what you'll see, the very top is plasma. It is the least dense component, so it settles on the top. The most dense components go to the bottom. And plasma is mainly made up of water. We talked about that, and that's 55% of the component of blood you're going to be your plasma. Also, the next coat is called the Buffy coat. Anybody know what the Buffy coat is? The white blood cells and the platelets will actually settle here in the Buffy coat, and it's that whitish, clear-looking coat. And sometimes we need to look at that particular coat, depending on what we're looking for. If we're a physician, or maybe even our MLT students might be looking at that. All right, and the very bottom layer is what? Red blood cells. The red blood cells and the Buffy coat together make up what we call the formed elements. Okay, and that's basically the cells, and red blood cells are the most dense, and they're in that bottom layer. It makes up 45% of whole blood, and this is called the hematocrit. And that can tell us, it should be a certain amount per volume. That can tell us something about that person. If you have too many red blood cells, not a good thing, right? Anybody remember from last semester what too many red blood cells could do to you? Makes your blood, and the proper terminology here would be viscous. It would give you, she said it would make your blood thick. It would make it highly viscous, sludgy, right? Another kind of slang term. And that viscosity is super important. Is viscosity important to a motor engine in a car? It is. Anybody ever put the wrong type of motor oil in your car? Would not be a good thing. It could destroy your engine. If you drive a Corvette, it probably will. And that's the same thing with your body. It's got to have the right viscosity to keep everything going as it should. So the blood has to have that viscosity. Too many red blood cells are a bad thing. Yes? Where? In Michigan. This is true. The viscosity is going to be dependent upon temperature and pressure. Y'all remember a little equation in chemistry? Anybody had chemistry? Pressure and temperature and volume and all those things are related. And temperature, it's kind of cold in Michigan, right? Also, sea level. What happens when you go skiing in Colorado? Have you ever done that? Anybody ever had altitude sickness? Come on, y'all don't ski. You should definitely make a trip to go skiing. It's a lot of fun. Before I haven't been skiing since I started having kids, but I used to love to go to Colorado. We would ski for 3 days and we would do nothing else. We would barely eat. We skied and skied and skied day and night. You go to Breckenridge, ski all day. You drive over to Keystone at night. You ski till pretty late hours of the night. I can't remember what the hours are, but anyway, fun trip. Second, I used to get on the mountain for the first couple of times, so I kind of got used to this altitude sickness. And it's just this immediate feeling comes over you of nausea, like you're not going to survive. It is extremely nauseating. What has just occurred is your body has just realized you're way above sea level, so what's the oxygen like there? Much less oxygen available. You have a certain amount of red blood cells floating around delivering oxygen. Not as much oxygen available. So you're going to start making more red blood cells. It's going to take a couple of days for you to generate more, but your body will start making more. Basically, you'll release a hormone called EPO, erythropoietin. That EPO will stimulate you to make more red blood cells. You can pick up more oxygen to drop off to the rest of the body due to decreased oxygen levels at a higher sea level. So yes, your viscosity is going to be affected by certain conditions. But within those certain conditions, it's got to have a certain viscosity. Yes? You can experience altitude sickness on an airplane. They are supposed to have mechanisms in place where they pressurize the cabin but I could see where something could go wrong there, and that wouldn't be exactly right, and you could experience altitude sickness. So I want you to understand and know what's going on here, because this is a basic technique. These MLT girls will be doing this technique. They're going to draw blood, and they're going to centrifuge it down, and they're going to separate the elements of blood in this fashion. So they're going to need to know what they've got and where they go to get certain things. And I'll let Evelyn teach you the rest of the story. But they will do that. And important to know if you're a healthcare professional. So you're going to be tested on that, right? Especially in lab. All right, let's go ahead and talk about the erythrocyte. What is the erythrocyte? That's basically the anatomical terminology for a red blood cell. So we could call them erythrocytes. I just referred to a hormone called erythropoietin, right? Erythro is telling you that's red blood cell production. Erythropoietin, that hormone is going to do that for you. Red blood cells are biconcave. When you look at them in a microscope, what do they look like? They look like little donuts because of that, and it's because they're biconcave. They're not actually a donut shape, but they're biconcave, so that's their appearance. In a human, they are the only cell that does not have a nucleus. Yes. Yes. Both sides look like, and you can see that from this picture here. That's a half of a red blood cell, and you can see that it's concave, biconcave. That's what bi would mean too, biconcave. So, no nucleus or organelles. So they have, basically they're bags of this protein called hemoglobin. They're little bags of hemoglobin. They contain a numerous amount of hemoglobin molecules, and I'll tell you about that a little bit later on. Their major function is the transportation of the respiratory gases, which explains why they are bags of hemoglobin. Because hemoglobin is going to be the molecule to bind these gases. Super important. They are produced by hematopoiesis. Hematopoiesis meaning red blood cell formation. You have specialized stem cells called hematopoietic stem cells in your bone marrow. If you'll recall, we talked about blood when we talked about bones, and this is the reason for that. Red bone marrow makes blood in general, okay? So we talked a little bit about that. So your red bone marrow is responsible for making your blood, and that contains hematopoietic stem cells to make the erythrocytes. And that, those cells last about 120 days in your blood. They have short lifespans because they don't have a nucleus or organelles. So they're not doing anything maintenance. They're 100% made and then function to transport gases. So there's no maintenance going on in that cell. And that's the reason why it'll have a short lifespan. Because they contain hemoglobin, after 120 days, that hemoglobin is what carries iron. So any, like when the cells, when the worn out red blood cells go off to be salvaged, like basically they're taken apart. We'll explain that process a little bit later. Our bodies try to salvage things out of them. Very efficient. So if there's iron attached to them, that iron is salvaged. But then the heme, remember hemoglobin, so there's a heme group and a globulin, which is the protein portion. The heme group is what actually binds the gases. It actually has an oxygen molecule in it in the middle that binds an iron molecule. So every heme will bind actually up to 4. Yes, Patricia. It has to do with they're not efficient yet at breaking down the hemoglobin, and there's a backing up mechanism that occurs. So they're not getting rid of the red blood cells to begin with. Most every baby born is going to have this issue. Usually their bodies kick in and start working through that. Sometimes they need a little extra help. but when this is not cleansed out of the body, it backs up and you can see it in the skin, pretty much. And that's why you have that yellowish tint. But the iron is salvaged. The heme is then degraded to a molecule called bilirubin. And then it's transported to the liver. So parts of this worn-out red blood cell will go into making bile. Anybody know what bile does? The liver makes bile. Let me just give you a hint. The liver makes bile. It ships it to a structure called the gallbladder. And your gallbladder releases bile into the intestines, actually into the duodenum, which is the first portion of your small intestines, coming out of the stomach. Anybody know what the purpose of bile is? Why do you... No. It's actually to help you absorb fat. Remember, your body is really polar, and fat is what? The opposite of polar. Non-polar. So remember, polar and non-polar doesn't mix together. So you got to have an emulsifier. Do y'all remember that term, emulsifier? What's an emulsifier? Y'all can't say breaks it down, and y'all got to get that out of your head. An emulsifier does not break down. An emulsifier is any substance that causes a polar and a non-polar substance to mix together. So it makes them compatible in a sense. So if you'll recall, what happens is you'll have, and the pen is not working or I would draw it for you, but what you have is a micelle forms from the emulsifier. If you'll remember, we're going to say this is the fat molecule, and it's nonpolar. If you'll recall, an emulsifier is a substance that has a polar end and a nonpolar end. So the nonpolar end, and remember in your cell, the cell membranes, you had nonpolar tails off the phospholipid bilayer, okay? So those little tails, typically a phospholipid, attach. They're attracted to because they're nonpolar, right? So they attach all around what's exposed. And what is the characteristic of the head? Polar or nonpolar? Polar. So what does that mean it can now do? And just imagine this little ball. It can now absorb into the polar surroundings. Yes. 100% exactly the same. And as a matter of fact, that's the example I use. And it's also the lab we do in Biology 1. We take soap. Soap is the emulsifier. We're going to do it here when we do digestion. We're going to make soap mimics bile in our lab. Soap is an emulsifier. When you wash your bodies off, contrary to popular belief, you cannot just get clean with water. Somebody needs to tell my little brother that. You've got to have soap. If you don't get soap, guess what's going to stay in your body? Probably the main thing you want off, which are the oils on your body, right? So an oily piece of dirt is going to stay, and a polar piece of dirt is going to wash off, right? So in order for you to actually get clean, you need an emulsifier, and that's what you're using. And way to think. Good job. So that's what's happening, and so bile does that. So some of the components from the breakdown of a red blood cell go into making bile. Now, when you go to make a red blood cell, so we're in your bone marrow, right? And we've got these hematopoietic stem cells. They're also called a hemocystoblast. They're going to go through a process, and you can see this process right here in this slide, where they will become a pro-erythroblast. The pro-erythroblast will become a basophilic erythroblast. It will go through ribosome synthesis. Remember, when the red blood cell spits out, there won't be any organelles, right? But we've got all that at this moment. We even see a nucleus. You see the nucleus? So the actual stem cell begins with a nucleus. So all of that happens here. We then have the polychromatic erythroblast, Phase 2, hemoglobin begins to accumulate. That cell starts to be made totally 100% into a bag of hemoglobin. You notice the nucleus getting smaller and smaller. The nucleus is actually spit out of the cell. It's ejected. In phase 3, the nucleus is ejected, and now the reticulocyte becomes the biconcave erythrocyte that you see. So that's basically the process of hematopoiesis. Nucleus is gone, salvaged for next time, or whatever happens. Whatever is needed for. The body will break it down, use components for whatever. Let's talk about the hemoglobin. So what did I tell you already about that hemoglobin? It binds respiratory gases. So this is what the molecule looks like. Does everybody see that? It's basically 4 protein chains. Do you remember me telling you the globulin part? When you hear the word globulin, think of a protein chain, because that's usually what a globulin is. So we've got 4 of these chains, and you can see them connected here. So there's 4, and each one of those chains has a heme group in it, a heme pigment. Each heme pigment can bind an iron molecule right there in the middle. So one hemoglobin can bind up to how many iron molecules? 4. Okay, so 4 molecules. Each hemoglobin molecule can transport 4 molecules. Each one of those will bind 4 oxygen molecules. So that iron group is what the oxygen binds to. So if you have low iron, for example, when you go to give blood, they're going to test your iron, right, to make sure you can't be anemic, and anemic means low iron. And that's because that would mean you wouldn't have enough oxygen transportation capability if you don't have the correct amount of iron. So each heme has an iron molecule, and it can bind up to 4, the whole molecule, the hemoglobin molecule can bind up to 4 oxygens, because it has those 4 irons in place. So the oxygen binds to the iron molecule you see in the center of the heme group. Does everybody see that? So this is the heme group. And it can bind an oxygen molecule right there. So up to 4 oxygen molecules can be bound to that group. We call that, when a hemoglobin molecule has oxygen bound to it, we call it oxyhemoglobin. Carbon dioxide binds on the protein chains as well. When CO2 is bound, we call it carbaminohemoglobin. So depending on what's bound to the hemoglobin, it can be oxygenated or it can be carbonated. Now, let's talk about leukocytes. Leukocytes are another word, or the actual anatomical terminology for what? White blood cells. So white blood cells is slang. It's actually known as a leukocyte. Leukocytes are the core of our defense system. Our immunity is dependent upon these white blood cells. They are our number 1 defense. They can actually leave the blood and move into the body. There's a process, and we actually talked about it, I believe, a little bit last semester in AMP1 called diapedesis. And it was, I think, when we talked about an injury, when an injury occurs, white blood cells actually go to that site. So they actually have to leave the bloodstream, and they do that through diapedesis, and those white blood cells go to the site or injury to do their thing. And what is their thing? We're going to talk about that, because they all have different things to do. So we're going to talk about the functions of the white blood cells. First of all, we actually separate white blood cells into 2 categories to begin with. We call them granulocytes and agranulocytes. This is sort of deceiving because they're named this way depending upon appearance, and appearance only. Because just because you call it an agranulocyte, meaning there are no grains, it means you can't see the grains. So it doesn't mean it doesn't have any. It's just when they stain, they are not visible. So keep that in mind. That's important in understanding the function, because it's called an agranulocyte, but it's labeled that way because it doesn't appear to have grains. That's a big important thing. The granulocytes, there are three types of granulocytes. I like to remember them as NEB. Neutrophils, eosinophils, and basophils. They are the granulocytes, and when they stain, they appear very grainy. Neutrophils are the most numerous white blood cells. They are going to be found in the greatest abundance, and that's because they're the bacterial layers. So when you think of a neutrophil, you think of the bacterial layers of the body. That's what they do. They go around and they get rid of the bacteria. They're very active in phagocytosis. So you have the most of these, why would you think neutrophils would be the most abundant? You have over 600 species of bacteria living in your body. Something's got to keep that at bay. Do you need bacteria in your body? Oh yes, absolutely. You do need those bacteria. That's why we don't always need to run to the doctor and get an antibiotic for everything, right? Because you take an antibiotic, what happens? You kill all the bacteria, good, bad, indifferent. All bacteria are wiped out. And guess what? Bacteria do a lot of things for us, and they keep a lot of things at bay. So important to keep your bacteria. What happens when we get an infection is some of the bacteria get out of control. Do you have staph on your body? You do. Some people carry it. Some people carry the bad staph. You've heard of the bad staph, MRSA. Your body's pretty happy as long as it's kept at an acceptable amount. And you'll learn in this class, it's not about the presence of something, it's about the amount. I've said that repeatedly in my classes. Because guess what, girls? You got testosterone in your body, right? Guess what, guys? You have estrogen in your body. If you're a girl and you have testosterone in your body, does that make you a boy? Because it's not the presence, it's the... If you have too much testosterone, could you start having some manly features? Yeah, you could. It happens. But we all have testosterone. It's the amount. If you don't have any testosterone, you're not going to have sex. That doesn't sound very fun, does it? Testosterone is responsible for your sex drive. So everybody's going to have some in their body. So it's almost never the presence of, it's almost always the amount. So bacteria can be present. It's just it can get out of control. And how do bacteria get out of control? It's when there's these wars going on all the time in your body and they're kind of fighting each other. Well, if the one that's usually fighting that bacteria, if something happened to them, then what's going to happen? That bacteria is going to flourish and take over and cause an infection. So that can happen if you take too many antibiotics. It can wipe out certain things or causing homeostatic balance to your body. So the neutrophils are the bacterial layers. They're going around making sure all that's happening as it should, and when they see the bacteria that doesn't belong, they're going to go attack. So you need lots of them because you got lots of bacteria. Eosinophils attack parasitic worms. So you'll see those. And the basophils contain histamine. Histamine is a vasodilator. It's involved in an allergic response. And this is an important part of what the basophil does because when it releases, it will attract other white blood cells. So that acts as an attractant. And you can see these here. So here's NEB, neutrophil, eosinophil, and basophil. Now let's just kind of look at the appearance of those three granular white blood cells. Remember, that's appearance. They look kind of grainy, right? The basophil looks extremely grainy. These guys, to me, are kind of easy. As a matter of fact, all three of these are kind of easy to identify typically. They are stained a certain color, and very often in A&P I say don't pay attention to the color. Color doesn't matter. I can stain it any color. But in this particular situation, color matters because I am staining it to get a certain color. And they're named because of that color. Neutrophils, which you see here on far left, neutrophils are neutral, and they're considered neutral because we are using an acidic stain and a basic stain, and they take up both stains neutrally. So what you see is the nucleus, pretty dark purple, but and the outside is kind of a light purple. It's taking up both the acidic stain and the basic stain equally. And usually the nucleus will have some lobes, as you see here. Eosinophils take up acidic stain the most. Acidic, you can see this in the pink outside. So first we see the grains. We're trying to look for grains. We see plenty of grains. And then we see the reddish color in that particular cell. Please note that this is much better to look at in your textbook, by the way. And I'll go ahead and point out that page number. 641. So if you look at page 641, you can see real obviously that eosinophil has that reddish cytoplasm because of the acidic stain. It also typically has a bilobe nucleus. So there's kind of three things we can look at. We can look for grains, and then we also look at the color, the cytoplasm, and we also look at the nucleus shape, and the fourth thing, the size of the cell. Size actually matters also. So we look at some different things in this identification process. And I will expect you to be able to identify these 5 leukocytes, these different types of leukocytes. I'll expect you to be able to know their normal compositions in the body, so the neutrophil being the most abundant, what percentage of the white blood cells are which one of these, and I also expect you to know what the function of each one is. So if eosinophils go up above normal, what might be the probable cause? Might have a parasite, right? Question. Is there a grain present? Does it look grainy in appearance? That automatically tells you granulocyte or agranulocyte. But that can also often be deceiving because look over here at the agranulocytes. They have a little bit of a grainy appearance also. But you have to look really carefully. You can see like distinct looking grains in a granulocyte, and these look kind of fuzzy. So you have to kind of become accustomed to looking at that. So is there a grain or not that you see? Second, let's look at the color of the cytoplasm. Third, shape of the nucleus. Fourth, size of the cell, because they do have different sizes. You'll need to be able to identify each one of these cells. Then you'll need to know, so what are normal values for these white blood cells? And then you'll need to be able to explain to me if one of them gets out of kilter, what could be going on. So what is the function of each one of those white blood cells? So let's look at the basophil. The basophil is pretty easy to denote because you can't even hardly see that nucleus. The nucleus and the cytoplasm are very difficult to distinguish. And also, basophils to me are almost the obvious at looking at grains. You can really see the grainy appearance in a basophil. But the basophil takes up more basic stain. So it becomes really purple. So the cytoplasm is almost the same color as the nucleus. And it usually has a bilobe nucleus, but it's very difficult to see the nucleus because it's so purplish black. The granules in it are very purplish black. So those are your granulocytes. Then we have 2 types of agranulocytes. The agranulocytes are your lymphocytes. Lymphocytes are broken down into 2 categories. You've got T cells and B cells. That's going to be very important when we get to chapter 21, I believe it is, called immunity. We will distinguish between T cells and B cells, but just to go ahead and throw it out there, T cells are called T cells because they originate in the thymus, and B cells are called B cells because they originate in the bone marrow. So T cells and B cells. We'll go into a lot of detail about those later on. Yes. T is for thymus, B is for bone marrow. And it's an agranulocyte. So we see our lymphocytes. You can also see in this picture here, that's one of those fragments that I was trying to show you earlier. That's a platelet, as you can see. Last one is a monocyte. Monocytes are what eventually are very abundant through the body and become macrophages. Macrophages engulf. Okay, they eat. They have voracious appetites, so they engulf and eat up things in the body. So they can actually leave the bloodstream as well, just like the diapedesis process I was talking about. So how white blood cells can actually leave the bloodstream. And you can see here, so the lymphocytes are small. These typically are pretty abundant. I believe that's the second most abundant white blood cell that you will find. They have a large spherical nucleus. So sometimes the nucleus almost takes up the whole cell. You can't see as much cytoplasm. So you can see that. Look how big the nucleus is, just a sliver of cytoplasm that we see. So they're a smaller cell, large nucleus, very spherical. The cytoplasm is like a pale blue. Monocyte is much bigger than the lymphocyte, and it always has this kidney-shaped nucleus, most commonly. Sometimes you see these out of whack, and they look pretty weird, but a common shape here is a kidney-shaped nucleus on the nucleus. So you can kind of see that. It also has the pale blue cytoplasm, but you can see much more cytoplasm in a monocyte than you can in a lymphocyte. So those are kind of some identifiers to help you to identify that. Also on page 641, you can see they've taken, let's see if we see that. That's not actually in our PowerPoint, but it is on page 641, and it's worth noting that you can see the abundance of the white blood cells. So you may want to go ahead and make a note of that, or highlight that, figure 17.9. And we're actually taking that leukocyte, the bussy coat, a majority of the bussy coats are your platelets. If we look at the leukocytes, it actually tells you, if you were taking a differential white blood cell count of a normal individual, what values you would find. I told you the neutrophil was the most abundant, and actually 50 to 70% of your white blood cell count should be made of those bacterial layers called the neutrophils. And I just told you lymphocytes were the next one, and you can see lymphocytes, and they're either T cells or B cells, make up 25 to 45% of that leukocyte layer in a normal, healthy individual. Then you have monocytes. 3 to 8% of your white blood cells will be a monocyte. Then behind that you have your eosinophils, 2 to 4%, and then basophils, 0.5 to 1% in a healthy individual. So if those values get out of kilter, it could be indicative of some type of pathogenic issue or a homeostatic imbalance. So we look to the blood to be able to read and tell different things about what's going on with a person. And basically, what is disease? Disease is any situation in which you go out of homeostatic imbalance, right? So the goal of the body is to maintain homeostatic balance. If your body gets out of kilter with homeostatic balance, then usually, typically, we have some type of disease. Platelets are not cells, so we don't consider a platelet a cell. Guess what a platelet is? It's a cell fragment. So no, there's not going to be nucleus or organelles because they're not even cells. They're just little fragments, and basically what happens is a megakaryocyte is breaking off little pieces to make up these little fragments involved in clotting of blood, right? So here's your hematopoietic stem cell in the bone marrow, and these cells are designed to become platelets. So that it will actually become a megakaryocyte, and megakaryocytes are going to become cells that release little bitty slivers of themselves, little fragments of themselves, and it enters the bloodstream as platelets. And these platelets are super important. They contribute to the viscosity, but they go to a site of injury to cause the clotting to occur when certain signals cause them to go to that site, and they'll actually start to adhere to the site and cause clotting to seal off that site of injury. So they make a temporary plug, and it clogs up the plug and we quit bleeding, right? Alright, so what does happen in an injury? The process is called hemostasis. So when you have a blood vessel that experiences a tear, certain things happen. First thing is vascular spasm. Directly where the injury occurs, platelets will release chemicals. You saw in that picture we just looked at, you can see little bitty chemicals inside those platelets. That's important because they will release those chemicals. Those chemicals are signals getting everybody involved that needs to be involved in an injury. So those chemicals release. Reflexes from pain receptors will lead to vasoconstriction. So that vessel constricts to reduce blood loss, right? So it's major vasoconstriction. Platelet plug formation occurs next. The platelets have been signaled to start to stick to these little collagen fibers that are exposed, right? So they're exposed, so they're now beginning to stick to them. Since they have been signaled to stick, they begin to release ADP, serotonin, and thromboxane. These are chemicals involved in the coagulation process, so all that's important. Coagulation then occurs. Fibrin threads will form around these aggregated little platelets, and they act like little cages catching the necessary components. Prothrombin activator is released, and that's activated. It's already in the blood, so this activates that. The thrombin combines with the fibrinogen to develop a mesh network. So that mesh is trapping red blood cells and platelets to form the clot that will seal off the point of injury. And that process is called hemostasis. That brings us to our blood groups, which we began talking about today in lab. Red blood cells have, if you'll remember, they have tags on them, right? Y'all remember what I called them? So they have basically antigens. So they have these present, or also known as an agglutinogen. They're little glycoproteins, glyco meaning sugar, protein, amino acid base, remember, that are hanging off the cell. They usually hang out of plasma membranes. All our cells have them. They identify us. They identify the cell. And these red blood, these ABO blood groups identify the cell with those particular groups. So they're called antigens or agglutinogens. That terminology is interchangeable, so we can use either one, an antigen or an agglutinogen. They're super important because this is one of the only cases that I know of in which you already have antibodies in your body to an antigen that you have never been exposed to. Earlier I said, how do you get antibodies? Through exposure. This is the exception. You are born with these antibodies. So if you get the wrong blood type from day one, you could die. It could be very bad. This is one of the only situations in which that would happen. Typically, you have to be exposed before your body builds up antibodies, right? That's how we fight off viruses and all kind of diseases. First exposure is pretty bad, right? You get the same strain of the flu twice? No. If you get the flu twice in one season, you got 2 different strains, and there's about 4 going around right now. The flu shot that's going around gives you immunity to about 3 strains. It's a 3-strain flu vaccination. My pharmacist told me that next year it will be 4. You'll be immune to 4 different strains of the flu. But who knows? We may have 8 different strains next year. It just depends on the season. So you have to build up immunity. So you won't get the flu twice if it's the same strain, because your body's already been exposed. It knows how to fight that. So if it knows how to fight the fight, it fights it immediately and you're good to go. Blood is not like that. We don't have to have exposure. Basically, if you are one of these groups, if you contain these antigens, whatever antigen you don't have, you will have antibodies to it. And that's why this particular antigen or agglutinogen is so important to us, because if you remember earlier, I said there's about 20 different antigens on your red blood cell. There are 2 that are super important for blood transfusion. And there's actually, if you have some type of issue where you have to have multiple or common blood transfusions, there's actually more to the story that you have to learn. You actually have to learn about those other antigens. But in this particular case, for the average person, what we're concerned with is the ABO blood types and RH factor. Guess what? RH factor is one of those where you build up antibodies through exposure. So it is different from the ABO antigens. Okay, so ABO, we're born with the antibodies. RH factor, we have to be exposed. And I'll explain that to you momentarily. But let's talk about ABO. So type A has the A antigen, right? Type B has the B antigen. Type AB has both. And type O, no antigens. Antigen-free, right? When we're talking about the ABO types. Every person has antibodies in the blood, and these antibodies can also be called agglutinins. So antigen or agglutinogen, and then we have antibody or agglutinin. And that's just terminology that's changeable. So these agglutinins that are found in the blood function to react to an agglutinogen. So they fit together, right? Type A has B antibodies. Type B has A antibodies. Type AB has no antibodies. And type O has both A and B antibodies. And this is your second time to hear this today, and you may have to read this 4 or 5 times before it sinks in. But it is important. AB, if you're AB blood, you can get any type of blood. Yes. And type O blood can only get O. So O is the universal donor, and AB is universal recipient. Very good. And that's important. Go ahead and denote that. So O is universal, and it's actually O negative when we talk about RH factor, and AB positive. Okay, so O negative will be universal donor, and AB positive is universal acceptor. And there's going to be questions for you about that. All right, so when we go to lab, when we do our lab, you'll see this. And when we go to lab, it'll show you if you have a reaction and you see coagulation and it'll look like little grains in the blood, that's the blood type you have. So you'll have an A place, a B place, and it's the same kind of blood. So you'll put the blood in the type A and you'll put the blood in the type B, and you'll have these little vials that are antibodies. You'll have antibody A, antibody B, and you'll have RH factor, anti-RH factor. And when you have coagulation, then you have, when you use antibody A, if you have coagulation, that means you have type A blood. If you use antibody B and you have coagulation, that means you have type B blood. If you have coagulation in both wells, that means you have type AB blood. No coagulation means O. So coagulation is telling you that the puzzle pieces fit together and cause clotting. Remember, only antibody A can fit into antigen A. So all of this can be very confusing. You can't memorize this. You need to think about it. You think about what's happening, and be able to think through this. And then you can figure it out. Yes, Alexis. Why does it clot? Remember how in lab I drew you a little picture of the antigen and I showed how the little antibody fits into there? Well, they fit together and clot, so they stick together. And first thing will happen, when you get the wrong blood type, you'll go blind because it'll stop, that clotting stops up the little bitty capillaries in your eyes, and then so much clotting can occur depending on how much blood you got, it'll start clogging up different arteries. Yep, means you got the wrong blood type. That means these ladies read it wrong. But they're not going to do that because they're going to be very well prepared because we have a wonderful MLT program. Not after a certain amount of time, that tissue will die if it doesn't have oxygen. You can receive A positive or A negative blood. I'm about to tell you about the plus and the negative are in regards to RH factor. Okay? These are little examples up here where you can just go through your mind as to what they could receive. Okay? So, let's see. If the donors, it's telling you the recipient. So if you're B negative, can you get AB positive blood? No. If you're AB positive, can you get B negative blood? Yes, you can. If you're A negative, can you get A positive? No. It's the plus and the minuses, and I haven't taught you that yet. I'm about to do that. If you're A positive, let me tell you what the plus and the minus are. The plus and the minus stand for RH factor. Plus means you have the antigen. Minus means you don't. Right? Here's the thing. The first time, no big deal. You can get the wrong blood type with regards to RH factor. Second time is the problem, because then you've built up antibodies to an antigen you don't have. Right? How do we build up antibodies? Exposure to a foreign antigen. So if you don't have the RH factor on your blood cells and you're exposed to it, your body will start to make antibodies to that antigen. So that's the reason why the plus and the minus are important. So let's think about this for a second. Plus means, yes, that the RH factor is present. So let's talk about that. I've only got like 3 minutes left, so let's hurry up and talk about that. It's really important in pregnancy when a mother is RH negative. Anybody in here had that? Anybody in here RH negative? If you're female, it's important. If you're male, we don't care, unless you need to get blood. But in pregnancy, it's really important because the RH factor antigen is dominant. So if dad has it, there's a good chance the child is going to have it, if you remember your genetics lessons, right? So if you're an RH negative mom and you get pregnant and the baby is RH positive, your blood doesn't mix until birth, right? At birth, your body is exposed to baby's blood, right? And it has that positive antigen. So what does your body start doing? Antibodies. You now have antibodies. Antibodies are easily transferred from mom to fetus, okay? That's one of the ways the fetus gets antibodies. And that's how they get some immunity when they're born. So while blood doesn't mix, antibodies do. So mom, who has been exposed to child 1's antigens, gets pregnant with child 2. That fetus, if it is RH positive, what's going to happen? The antibodies that cross are going to attack the fetus, right? So then the fetus will not be able to survive. So what they do is they actually give a immunosuppressant shot to mom at the end of towards the end of the last trimester, and I think actually at birth, that keeps mom from making those antibodies so that if she decides to get pregnant again in the future, she won't have those issues. So that's where RH factor comes in. In that case, that situation is called erythroblastis, I believe, fatalis. It's in your textbook. It is important. I want you to read it. I want you to know it. But in regards to RH factor, so basically, the plus and the minus tells us about RH factor. And so a universal donor is going to be O negative. Universal recipient is going to be AB positive. Do you all understand why? Read this, pay attention, and think about what I've told you. And then come back on Monday and we'll have a conversation to see if you've got it. Okay? Now, you need to read this chapter. Blackboard is not ready for you to do the enhancement assignment. I'll send you an email as soon as I get that going for you. Tomorrow I will be in Jackson. So if you need me,